Hello, I am Dr. Jean-Claude Manuguerra. I'm a virologist and unit director at the Institut Pasteur. I run the Laboratory for Urgent Response to Biological Threats, and we're proud to have participated to the design of a saliva-based rapid test for SARS-CoV-2 diagnosis, further developed and CE marked by C4 Diagnostic Biotech Company. Before diving into the clinical performance of that test, I shall tell you a bit more about our group at Institut Pasteur, browse the current landscape of available tests for COVID-19 diagnosis, and how we leverage on our expertise to design primers for robust and rapid detection of SARS-CoV-2 virus and its variants. The Institut Pasteur was established in 1887 with 4 missions at the service of scientific progress and the improvement of public health. 1, research, 2, public health, 3, education and training, 4, innovation. Its Laboratory for Urgent Response to Biological Threats was established in 2002 to respond to specialized biological emergencies that can endanger public health during epidemics, accidents, or bioterrorist attacks. It has 2 modes of intervention. The direct mode, where it acts as an independent laboratory upstream of the national reference centers, conducting independently teaching interventions during epidemics and research, as well as giving advice. For this, it can use its collection of viruses, bacteria, and recombinant antigen. It also has an indirect mode, and in this case, it provides logistical support in human and material resources to other laboratories overwhelmed by their workload. It is very important to detect carriers of the SARS-CoV-2 in the population in symptomatic people, but also in asymptomatic people. Symptomatic people must be tested to alert, trace, protect, but first to treat these infected people, also to isolate them or hospitalize them if necessary. In addition, it allows to trace and isolate the contacts of these patients with COVID-19. It is also important to test people without symptoms. There too, to alert, trace, and protect. This is particularly important in certain contexts such as schools, sporting events, gatherings, travels, for example. Indeed, more than 50% of all the transmissions come from transmissions by people without symptoms, which come from 35% of pre-symptomatic people and 24% of individuals that never develop symptoms despite their infection. There are several categories of virus detection tests. First, we have real-time RT-PCR, whose detection limit is 10 copies per reaction, which serves as a gold standard for sensitivity and specificity. The reaction time is 90 to 120 minutes long, and the necessary instrument is expensive. The other technology is real-time RT-LAMP, which has a detection limit comparable to that of RT-PCR, a sensitivity of more than 95%, and a specificity of 100%. The reaction time is much shorter, under 30 minutes, and the necessary instrument is much, much cheaper. Finally, there are the antigenic tests, the detection limit of which is much higher, on the order of 1 million copy equivalent, and therefore its sensitivity is much less good, at best 60 to 80%. Its specificity is good at 90%, and the reaction time is 15 or 20 minutes, and most often no instrument are needed. Our choice, therefore, fell on the RT-LAMP. For these tests, it is possible to work on different matrices: nasal, pharyngeal, nasopharyngeal swabs, or saliva, which is an excellent biological fluid given the presence of SARS-CoV-2 in the salivary glands. This RT-LAMP was designed on 2 different targets located in the RDRP and in the N genes, which are 2 conserved regions of the genome where there are very few mutations, unlike other regions. Mutation may impact the sensitivity of the tests. In fact, most of the important mutations are observed in the S genes coding for the spike. These mutations are the main characteristic of the variants. The test was developed so that the specific primers of the RDRP gene recognize the SARS-CoV virus and the MERS virus. The primers directed against the N gene are specific for SARS-CoV-2. The test was verified using cultivated virus of SARS-CoV-2, SARS-CoV-1, and MERS-CoV. For SARS-CoV-2, we used the classical virus as well as the English, Brazilian, and South African variants now called alpha, beta, and gamma. In addition, an in silico verification was carried out in May 2021 by blast analysis on sequences obtained from isolates that had circulated in France during the last 6 months. That's to say, more than 65,000 sequences.
Hello, I'm Emilie Fugier, Head of Technology and Innovation Department at C4 Diagnostic in Marseille. I will present you the clinical validation of the RT-LAMP SARS-CoV-2 test called C4Covid-19 Human, which is a test for SARS-CoV-2 detection and diagnosis. Its key features are a rapid detection of the SARS-CoV-2 in 30 minutes from saliva sample. The test is based on RT-LAMP technology that target 2 genes thanks to sets of primers designed and patented by Dr. Manuguerra's team. One gene is common to beta coronavirus family, and the other gene is specific to the SARS-CoV-2. In collaboration with Synapse Provence, we performed the clinical validation of our C4Covid-19 human tests on saliva in comparison with the gold standard, which is nasopharyngeal samples analyzed by RT-PCR. The comparative study occurred from February to April 2021. We included 1,320 subjects according to the following criteria: adult person with COVID-19-like symptoms or being contact cases and not having drinking, eating, or smoking 30 minutes before the test. The study design was the following. Saliva samples recovered in site was immediately analyzed within 30 minutes by RT-LAMP. Meanwhile, nasopharyngeal sample of the same subjects was analyzed within 24 hours by RT-PCR in a clinical lab. Here are the clinical performances of C4Covid-19 human test, that is now CE marked. We get 86% of sensitivity, 97% of specificity, and 95% of matching results. It has to be noticed that more than half of the subjects included in the study presented no symptom. So, C4Covid-19 human test detects symptomatic and non-symptomatic subjects. In addition, thanks to variant screening identification performed by Synapse on nasopharyngeal samples, we clearly demonstrated that C4Covid-19 humans is able to detect SARS-CoV-2 variants. The graph here on the slide represents the distribution of SARS-CoV-2 wild type and variants among matched positive saliva and nasopharyngeal samples. In conclusion, C4Covid-19 Human is a rapid and reliable test for SARS-CoV-2 detection. It works on saliva sample, it is compatible with variants, and it offers a level of performance comparable to those of the gold standard. Currently, this clinical study is still ongoing by including another analysis of saliva by RT-PCR in order to refine performances of the C4Covid-19 human tests. Final results are expected at the end of this month. To finish, if you want more information about our test, feel free to contact us. Thank you.